Well, thank you everyone for joining us today for this really important seminar. My name is Angela Colantonio, and I am a senior scientist at the Kite Research Institute at the Toronto Rehabilitation Institute at UHN. And I'm also the director of the Rehabilitation Sciences Institute at the University of Toronto and a professor of occupational science and occupational therapy. And I was asked by the UHN IDEA Committee to moderate today's session today, and I really thank them for organizing this session on an important topic. I am of European settler descent, born, raised, and living in the unceded territory of the Mississaugas of the Credit, Anishinaabeg, the Chippewa, Haudenosaunee, and the Wendat people. And I want to start this session by first acknowledging the land on which UHN operates. There has been a UHN hospital on this land since 1829. Long before that, Indigenous peoples were its stewards. They, as their descendants, and we at UHN do now, learned, shared knowledge, and healed each other. Regardless of where we sit today, we owe much to the peoples that have called this land home. For thousands of years, it has been the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabeg, the Chippewa, the Haudenosaunee, and the Wendat peoples. We also acknowledge that Toronto is covered by Treaty 13 and the Mississaugas of the Credit. This land is now home to many diverse First Nations, Inuit, and Métis people, and we are grateful to have the opportunity to live and work here. We remember and honor the legacy of the peoples who have been here before us, and all who work to make the promise and the challenge of truth and reconciliation real. I would like to invite you to join me in taking a moment to reflect on the land that you are on. I would now like to introduce Dr. Ritz, who will be speaking on the integration of sex and gender in research, new advances and important considerations for basic and clinical research. As in terms of her bio, Dr. Stacey Ritz is an associate professor in the Department of Pathology and Molecular Medicine at McMaster University. After completing her Ph.D. in immunology at McMaster and a postdoctoral fellowship at UCLA, she served as a faculty member at the Northern Ontario School of Medicine from 2005 to 2015, and returned to McMaster in 2015 as the Assistant Dean of the Bachelor of Health Sciences Honors Program. Her current scholarship focuses on the integration of sex and gender considerations into laboratory-based biomedical research and health research more broadly, and critical literacy in health professional education. So thank you for joining us today, Dr. Ritz, and we look forward to your presentation.
Thanks very much, Angela. Thank you so much for the invitation to join you today and to talk about this work that I feel so excited and passionate about. As you mentioned, my background is as a laboratory researcher. I did my PhD looking at the cellular and molecular mechanisms of allergic sensitization in the respiratory tract and in using animal models of allergic disease. And then when I established my own research program, I was also looking at mostly animal models and cell culture models to look at the impact of air pollutants on the immune response. And I also came to that work with a deep interest in feminist perspectives on science and an interest in how do we address sex and gender considerations in this kind of work. I started my career just as just as the CIHR itself was being formed, and we had a new Institute of Gender and Health, and I was lucky enough to be invited to a number of the early meetings of the Institute of Gender and Health to sort of set priorities and think about how the Institute could foster more integration of sex and gender considerations in research in the Canadian context. And so for many years, that work, that impulse and impetus to integrate sex and gender in health research was very much in my mind, but sort of off to the side and in the back a little bit. And in the early 2000s, as this was particularly becoming more and more prevalent, more and more heightened priority for many people, the desire that I had to think about how to do this was I was finding a mismatch between a lot of the direction or the policy or the recommendations that were coming from some of the funders, some of the other organizations, a mismatch between what they were asking for and what was feasible in my laboratory-based work. So probably the most significant example of that was, people saying, well, if you're doing cell culture research, you need to include cells, both male and female cells in your work. And I was just, well, like that just doesn't work for people who are doing, transformed cell lines. There's no way to get a female A549 cell line, and there's no way to get a male HeLa cell line. And so I just felt this massive disconnect. And over the course of a number of years, that gap became my obsession and my passion. And I began to focus more and more of my work thinking about those kinds of questions, and ultimately wound down my laboratory-based research to focus more on taking up these kinds of questions. And that's what I've been focused on now for probably the last 10 years or so. This has become my primary focus. So in the Canadian context, probably most Canadian health researchers are pretty familiar with the distinction between the concept of sex and the concept of gender. We've been conditioned to that in the last probably 13 years, I think, since the CIHR began requiring all applicants for CIHR funding to answer questions about how they will address sex and gender considerations in their work. And the exact nature of those questions has adapted over the years in response to program evaluation about how researchers were taking those up and how best to probe those concepts, how best to integrate them into the review process. So I'm not going to belabor this too much because I think most people are pretty familiar with it if you're in the health field in Canada already. But if we think about the distinction between the concepts of sex and gender, there was a time not too long ago where people used these terms interchangeably. And in fact, sometimes they would use gender, I think, more often because it sounded less sexual. And so gender would get used when they really meant sex, or they were used interchangeably. But I think that is less now than it used to be, partly because of this heightened awareness through the CIHR. These particular definitions are ones that that Lorraine Greaves and I have proposed in a recent paper that we collaborated on together. And, it's really kind of an integration of our thinking about this over many years from many different sources. And I'm never entirely happy with them, but this is sort of where we've settled provisionally for the moment. And they're largely in line with the CIHR definitions, where sex is understood to focus principally on things related to biological attributes and functions, and gender is principally conceptualized around aspects of social, cultural, political organization that shapes experience, norms, behaviors, aesthetics, relations, and so on. A couple of things I want to highlight here, though, is particularly around the question of gender as big gender, as opposed to focusing entirely on gender identity. I think one of the things that is interesting and noteworthy and generally quite positive is that there's been a real heightened awareness socially, culturally, in the broader public discourse about gender norms and gender roles and gender identities outside of the, very conventional, hegemonic, normative male, female, cisgender binary. And I think there's been a whole lot of really positive, terrific things that have come out of that and will continue to move in that direction. I think one of the things that I find I often want to highlight when talking about gender in the context of integrating gender considerations into health research is that when we are talking about gender in this sense, we are talking about gender identity, but we are not only talking about gender identity. We're talking about gender identity, but also gender roles, gender relations, institutional and systemic aspects of gender. And all of these different things can intersect and influence one another, but we're not only talking about identity. And I often find that important to highlight, especially for people who are working with animal models and working in experimental settings where they're like, well, I know the external genitalia of my mice and can assign them that way, but I don't know if they have what their sense of gender is, if mice even have one, et cetera, et cetera. And I think, I often have to offer a clarification there that when we're talking about gender, we're talking about more than simply identity. And there are some important ways I think we can think about that even when we are working with mice or even when we're working with cells. The other thing I wanted to highlight is that I think this distinction, this conceptual distinction between sex and gender is important and it's useful. But the other thing to highlight is that in practice, they are not really as neatly separable as these definitions are often interpreted to be. They are in constant dynamic dialogue with one another. And sometimes it's very difficult to discern whether we're talking about something that is properly called sex or whether we're talking about something we should be calling gender. And sometimes it's both. And so increasingly, I've seen people making use of the hybrid term gender/sex or sex/gender as a way of acknowledging this entanglement. And I think I'll probably do that sometimes in this talk. And that's what I'm trying to acknowledge and evoke when I do so. The other one thing that I think is a bit problematic about this distinction, this very clear binary understanding of sex and gender, is that it tends to lead people to thinking about sex and gender as if sex is in the body, gender is outside the body, and that there's a neat boundary between those things. When in actuality, there's a lot of dialogue and dynamicism between them. I really like this quote from King and Malin a number of years ago, where they're talking about sex, but I think it's actually about sex and gender. Is that this is an ongoing process. It's not one fixed, category that is fixed and static and binary throughout one's entire life. Sex and gender, I would say, are ongoing processes, and our bodies are open to the planet. There is, we do translate all kinds of our external experiences that we might call gender, get translated into biology, into our bodies. And Lorraine Greaves and I have talked about this a little bit in our recent paper, talking about some of the ways that we can think about how gender gets into the body and gender can become biological. And this is not an exhaustive review. It's really just meant to highlight some potential examples. So things that are related to gender in many cultures, for example, things like clothing styles, can become translated into biology with potential health impacts when you think about how they mediate different types of biological and physiological functions. So being more or less covered and using or not using sunscreen can translate into biology, for example, by modulating the amount of sun that we're exposed to, and therefore our endogenous vitamin D production. And then vitamin D levels can go on and have a number of different potential health impacts. The kinds of shoes that we wear influence our risk for musculoskeletal pain and malformation that are absolutely related, translated through biological pathways. Occupational and domestic roles can influence our exposure to different kinds of risk factors for different health problems, different diseases. Gender norms of things like toughness or gender norms related to risk-taking can be translated into biology related to things like underreporting of pain and so on and so forth. I'm not going to go through all these in detail. One I did want to highlight, though, is about the ways that gender can be translated into biology through the socially mediated modulation of steroid hormone production, and particularly testosterone production, because I think that is more well studied than some of the other ones. We have a lot of cultural narratives about testosterone and masculinity in particular, and especially the idea that testosterone is the cause in certain ways of certain aspects of cultural masculinity, related to aggression or sex drive or nurturance and competitive behavior and that sort of thing. So we tend to have a cultural narrative of this one-way causal pathway that testosterone causes masculinity in these ways. In fact, there's a lot of interesting research that shows that the inverse relationship there may actually be more evidence and more support for the premise that our behavior and our experience in the world influences androgen and testosterone production. So, for example, it's well documented that behaving in competitive ways, behaving in aggressive ways, behaving in ways of wielding power, like firing people, increases our expression of testosterone. Other kinds of behaviors, like engaging in nurturing behaviors, decreases our testosterone production. So it's not as simple. There's a more complicated relationship between the biology and the social, I think, than we often think about and give credit to. So I've talked now about I first talked about the ways that sex and gender aren't really a clear-cut division between sex and gender and the way that they talk to each other. I also want to talk about the other big binary when we're talking about sex and gender, which is the male-female binary that is used as a framework for thinking about sex and gender very often, and I'm going to argue way too often. I think there are lots of things that are important to scrutinize and critique about this, but I think some of the most substantial ones for thinking about health research and how we design and analyze health research studies are to recognize that sex and gender categories are not clear-cut, well-defined, absolute binaries, and that no single trait defines a sex or gender category. Sex and gender both are better understood, both biologically and socially, as constellations of a large number of traits. And we often find that these are correlated or associated with one another in certain kinds of clusters, but these clusters aren't absolute, and they certainly aren't simple binaries. And the other piece to highlight is that many of the traits that we tend to associate with maleness or femaleness are not static. Many of these are quite dynamic and change both acutely and over the whole lifespan. So, for example, one of my pet peeves, I guess, is talking about hormones, talking about steroid and gonadal hormones as if they're male hormones and female hormones. That, estrogen is often described in many textbooks and a lot of literature as a female hormone, which is, actually inaccurate. It's literally not true. All bodies have and need estrogen for normal function. And the idea that it's a female hormone is problematic. Additionally, because for huge swaths of the life cycle in humans, estradiol levels are actually very, very similar in males and females, both in childhood, postmenopausally, and even when women are in estrous or menstrual cycling and have much more varying levels of estradiol, there are absolutely times during the menstrual cycle where an individual's estradiol levels are very comparable to those typically seen in men. So the idea that it's a female hormone is problematic in a lot of ways. The other big way that it's problematic is that when we restrict our thinking about sex and gender to, okay, I'm going to compare males and females, doing so without any further thoughtfulness or critique ignores the reality of human heterogeneity, diversity within those categories, the realities of people who don't identify with or do not fit those categories. And like I said, the dynamic nature of a lot of the traits associated with sex and gender. Again, a lot of this kind of comes to our cultural narrative about the opposite, thinking of men and women as the opposite sex, or I think it was the men are from Mars, women are from Venus phenomenon, if you remember that book from the 90s. This idea that men and women are so different from one another that they are actually from different planets. And when we confront it, I mean, I think most people are like, haha, obviously not. But actually we talk and behave and think and analyze oftentimes with that idea in mind. But as Harriet Lerner, one of my favorite authors, has written about this, when we think about men and women as a binary, an opposite binary, men are from Mars, women are from Venus, that has two big problems. Number one, it obscures the huge commonalities that we see across all of human existence, but also obscures the range of differences that are very normal within each of those groups. And I've just included this graph here showing height as a very common sense kind of example, where we think about, like the broad umbrella brushstroke about height and gender is that men are taller than women. But we all know that not all men are taller than all women, and that the distributions for height overlap considerably for men and women. But when we say men are taller than women, we are saying the mean height for men is larger than the mean height for women. But we do recognize that there is this overlap of distributions. And in fact, that is the case for just about any trait that we think of as being associated with sex or gender. If you look at these different types of distributions, there are a handful of characteristics we associate with sex and gender that have a fairly distinct distribution. But those are actually quite rare. And even there's almost always some overlap between the distributions. Much more common are distributions that look like these other forms, where yes, we may see some mean differences between the categories of male and female. And those might even be statistically significant. But when you actually look at the distributions and you see what the range of individuals for whom the data or that feature is in an overlapping territory is very, very broad and very, very common. And so I think it really behooves us to remember that when we're talking about males versus females, we're talking about sex differences, when we are using that framing, that we are in fact usually talking about mean differences, not binary opposite kinds of differences. And we see that come up in research and scientific discourse all the time. So I want to highlight 3 kind of case studies about why a simple male versus female, men versus women kind of approach to incorporating sex and gender in research falls short and fails us for really trying to understand how sex and gender related factors influence our health. And the first example is one that's quite an old one, but it's one of my favorite ones. It's one of the examples that was one of the first that drew this phenomenon to my attention, actually. And it's work from ergonomists in Montreal who were looking at the prevalence of a shoulder injury in mechanics working in a diesel engine shop. And there was a particular shoulder injury that was much more common in women mechanics. Now, there were very few women mechanics in the shop. There was only a very, 3 or 4 of them in a shop of, 150. But almost all of the women, I think, had this shoulder injury, and it was an injury that was seen in some of the men who were mechanics, but at much lower rates. And when they actually sent in an ergonomist to understand what was going on, it became fairly clear that the risk factor here wasn't actually being female, even though the injury was much more prevalent in women. The problem was the work angle at the shoulder was meaning that people who were shorter had to reach up and exert force where their arm was elevated above the level of the shoulder more often. And that action was creating I'm not a biomechanics person, but was creating some tension on the joint that what made them more susceptible to this injury. And so it was very clear from the analysis that the issue here was not femaleness at all. It was height. But because women are more likely to be short, it was seen with a higher prevalence in that group. But it was not femaleness or anything about being female per se. It was simply about height. And I especially like that example because unlike some other examples, many other instances, there's really not a hormonal explanation for this either. It's really just simply about height. A more recent example, a few years ago, there was a study done of Canadian blood donors, looking especially at people who were frequent blood donors. And what they found was that women who were frequent donors were much more likely to have low ferritin levels than men who were frequent donors. So they had done their analysis looking at sex category. They had done some different age groups in the female group, and they had looked at donation frequency. But a number of the factors they hadn't considered were things like menstrual losses, body size, dietary iron intake and vegetarian diets, or any other factors that were associated with sex and gender. So I found this to be problematic because when you think about what the practice of blood donation is, they take a pint of blood from every donor, regardless of body size, regardless of menstrual history and so on. And the differences that they saw between women and men who were frequent donors could very well have been explained mechanistically by things like, the extent of menstrual losses of iron, by body size, by dietary iron intake. And what they ended up doing on the basis of these findings was in order in an effort to protect the health of women donors, they changed the interdonation interval to 12 weeks instead of 8 weeks, as it had been previously, but they left the interdonation interval at 8 weeks for men. And as well-intentioned as that was, I think one of the big problems with that is that this basing of policy on sex category alone, without any consideration or integration of understanding about possible mechanism, functionally means that they treat two women. For example, you could say, one donor is 60 kilograms, has heavy menstrual losses, and is a vegetarian, with a donor who is, postmenopausal, has no further menstrual losses, who's 5'10" and 100 kilograms and eats meat regularly. It treats those two individuals as if they are in the same risk category simply based on their sex category, their gender category. And it treats, that larger postmenopausal vegetarian or meat-eating woman as if she's at higher risk than a 60-kilogram vegan man. And so it's based a policy intending to help people on something that has nothing to do with the mechanism. And so we're misdirecting our efforts to protect health. So there are women who don't need this protection who are getting it, and there are men who are at risk who are not being protected by this policy. The other thing I would note is that, when they looked at their most frequent donors, people who gave 4 times a year or more, there was about 20% of the women who had totally normal, sorry, this should read ferritin, not hemoglobin, who had totally normal ferritin, and only about 36% of men who had normal ferritin as well. So again, that's just evidence that they are basing a policy on sex gender category instead of mechanism is not actually orienting the strategy to mitigating risk in a very effective way. The final example I want to offer is a very recent one about looking at sex and gender in COVID-19. In this paper, Takahashi et al., this is a Nature paper, looking at immune responses in COVID-19, comparing men and women. They found statistically significant differences in several immune parameters, including IL-8, IL-18, and T-cell activation. And in the abstract and in the paper, on the basis of these findings, they say not only does this provide a possible explanation for the observed sex biases in COVID-19 outcomes, but they also propose that their findings provide a basis for the development of a sex-based approach to the treatment and care of male and female patients with COVID-19. When you actually look at the data for these parameters that they had found statistically significant differences in, the wisdom of a sex-based approach is called into question. If you look here on the Y or the X-axis, the left 2 groups are healthcare workers who were not infected with COVID-19. So these are the controls, male and female female patients. And for IL-8, where they said, this was a significant difference. Yes, the difference between male patients and female patients in their expression of IL-8 and IL-18 was statistically significant. But when you actually look at the distributions of the data points for the individuals, you can see that for most males and most females were actually very similar in their expression of IL-8 and IL-18. There was only, a few at the higher end that were pushing the means up for men here. And if you look here at this is the T-cell activation. Similarly, the vast majority of both male and female patients had T-cell activation levels that were very similar to one another. It's only a relatively small number of patients who were dragging this up to become statistically significant. So when I look at these distributions, I don't see a strong argument for sex-based approach to the treatment and care of male and female patients. The argument I see here is very much most male and female patients are very similar to one another. But I think this is an example of the ways in which our men are from Mars, women are from Venus, cultural narratives about the opposite sex and that men and women are so different filters into our analysis and interpretation of data in ways that amplify and overemphasize difference when really the differences are not as pronounced as our rhetoric makes them sound. So how do we move beyond this male-female binary? What can we do as health researchers to try and transcend and move past it? I think for a lot of reasons, I think it will be difficult to do away with a male-female binary entirely, to do away with male-female comparisons entirely. Particularly, I think that's especially challenging with animal research. But I do think there are ways that we can moderate and be more thoughtful and critical about our use of a male-female binary. And so I'm going to say a few things about how we can rethink our data visualization, our statistical analyses, and our rhetoric about sex differences and male-female comparisons that will help us to reduce our this men are from Mars, women are from Venus kind of approach. But I also think we do have opportunities to start to shift our thinking about sex and gender, not simply in terms of male-female comparisons, but thinking about what are the sex and gender related factors that we need to be focusing on to help us understand the mechanism, rather than thinking about sex category. And I'm particularly going to lean here on some recent work by Sarah Richardson about sex contextualism, and I'm going to sort of tweak that and twist that into thinking about gender contextualism as well. So in terms of data visualization, sorry, these are a little fuzzy for some reason. I hope you can see that okay. This is it's 4 different ways of visualizing data from a male-female comparison that we've come up with just as an example. This is fictional data about a fictional protein. It's really just for the as an example. But if you look in figure A here on the left, this is what I see most often in the literature in terms of visualizing data where people are comparing males and females or men and women, where there's a bar showing the mean, there's a standard error, or in this case, we're showing 95% confidence interval, and there's some indication of the statistical significance. And so for this fictional protein that we've called protometaglobulin, you could see that the mean value for females here was 10 plus or minus, whereas it was about 7 for males. If you plot it this way, this most conventional way, the visual impact, the visual narrative of this really tends to focus and hyper-focus your attention on the difference between males and females. And that's conveyed both through the bars being different colors, through the bars just focusing and drawing your attention to the mean as the most important thing, and the emphasis on the statistical significance. But if you think about other ways to visualize that data that allow us to better appreciate the distributions, appreciate the overlap and the heterogeneity, you can see a few different variations that we've offered here. In B, we are looking at the same bars and the same error bars, but we are actually showing an additional point for each individual. And that alone, you can start to see, okay, actually, there's actually a lot of overlap here. In C, we've pulled out the bars. And so we no longer make this visual narrative of difference. We don't have black for male and gray for female. We're not conveying difference even in the visual itself. But by taking the bars out, we further reduce the emphasis on the means and place a greater visual emphasis on the distributions. And on the right, this way of presenting the data, the mean is still present. If you want that information, it's still there in the figure legend, but the visual message here is much more one of overlap, similarity, and diversity within each group. You can see that within both the male group and the female group, there are, individuals at the low end and individuals towards the high end. And yes, there's a mean difference between them, but the vast majority of individuals are very similar. So when we offer these as examples, we're not trying to be prescriptive and say you should present your data in this way, but by thinking about what is the visual narrative that you are communicating, what is the discourse that you're perpetuating in your choices about data visualization, can help to shift our thinking about sex and gender. And I do want to add that even as I critiqued this paper, I do give them points for presenting their data in this way that did allow the reader to go in and make these appraisals for themselves and draw that conclusion for themselves. So that's, I think that is something to be noted as well. In terms of statistics, I am not a statistician. This is not an area of strength for me, so I'm not going to go into detail on this. But I think there is some evidence that the statistical analyses that people are using may be leading to an over-reporting of sex differences in the literature. And I'm particularly leaning here on some recent work by Garcia Cifuentes and Maney, where they looked at a collection of several hundred articles from the biomedical literature that had done a male-female comparison. And when they looked at the articles that claimed to have shown male-female difference, they found that 71% of those papers had either missing statistical analyses, or they'd misinterpreted or otherwise used inappropriate statistical analyses. They said that, for most experimental settings, if you are comparing male and female treatment groups and male and female controls, in most settings, the most appropriate statistical test would have been a two-way ANOVA to look at the sex by treatment interaction. And the majority of the papers they looked at had not used this test, and they had not used an appropriate alternative either. They'd mostly used a one-way ANOVA, for example. Shockingly, when they looked at the authors who did not test for the interactions appropriately, they were 7 times more likely to claim sex-specific effects than those who did the appropriate analyses. So that's quite alarming, and I think heightens this sensibility that we have about, men are from Mars, women are from Venus, and that these are heightens the narrative and discourse of difference. I think more broadly, even thinking beyond their particular analysis, when we think about a lot of the tests that are applied, especially in experimental research like t-tests and ANOVAs, we have to remember that statistically speaking, they are intended to detect a difference between groups. That's what they are trying to do. They're not oriented to discerning relationships or identifying complex interactions or looking at multiple factors. And so I think it behooves us to think carefully about what is this test designed for, and how are what are the implications of using it in this way? I'm just going to skip to the third point because I think it links a little bit more careful, or a little bit more directly here. I wonder, again, not being a statistician myself, but I wonder if there's some scope here for innovation in biostatistical analysis. Are there ways that other forms of analysis that are better geared to discerning relationships, looking at complex interactions and clusters, could be adapted to this kind of research? I would be very interested if there were people who had expertise in that area who were interested in developing some of those ideas. But the other piece I want to highlight here is that a male-female comparison is not the same as a comparison of most other variables that we're looking at, especially in experimental work. When you think about experimental design and control and intervention, the whole power of experimental design is to be able to hold all variables constant except for the single variable that you want to manipulate and look at the outcome. And that's where so much of the epistemological power of experimental work comes from. But a male-female comparison is not the same as comparing control versus intervention. When you are comparing a male and female, that sex category, gender category is not a simple singular variable. Male and female sex and gender categories are complex constellations of multiple things that are in dynamic, changing dialogue with one another. And so when you are comparing male and female, it's not like comparing placebo versus drug. It's like comparing placebo versus, making 2,700 different changes. The challenge is that, it's not like we have a whole lot of latitude to intervene in sex and gender the way that we intervene in our other forms of intervention. So it's not that there's an easy alternative to doing this, but I think it's really important for us to have a heightened conscientiousness that what we are doing when we're comparing male versus female is not the same as when we're treating, it's not the same as most other variables. I also want to talk about rhetoric. Some of this comes out of the Maney and Garcia Cifuentes paper, but some of it comes from other work that we've been doing and other work that others have been doing as well. A lot of times when people are doing male-female comparisons, they tend to use language to describe their findings like sex-specific, sex-dependent, sexually dimorphic, sex difference. I think it's worth asking if these descriptions are truly justified by the data, or if, again, our cultural narratives about male-female difference are really driving us to describe the data in these ways. So we've offered a few examples of how one might change the language, change the rhetoric that captures the description and interprets the data appropriately, but without simply relying on this narrative of male-female difference and explicitly acknowledging the diversity, heterogeneity, and overlap of distributions that we see from most of the data in these areas. And the first two examples in the table sort of illustrate those ways of shifting our rhetoric. The third example is a little bit different. And it's one that I've been finding especially useful in my own thinking and my own work in this area in the last number of years, is to shift my frame of reference. Instead of talking about the influences of sex and gender on health, I've started trying to think about the influences of sex and gender related factors on health. And I think that can be a really helpful shift in our framing, because it directs our attention to mechanism. It directs our attention to the causal pathways themselves instead of to these relatively arbitrary categories. And I think it will result in better hypotheses, better design, and better analysis if we shift our thinking to the factors themselves rather than the categories. Just keeping an eye on the time, and I'm almost done. The other thing I think we really need to be conscientious about is the ways that our cultural narratives and stereotypes about sex and gender influence our science and the interpretation of data and the design of experiments. And there's a number of really great examples of these about the ways that narratives and stereotypes about gender influence neuroscience. Here, this book, Delusions of Gender, influence work about testosterone, influence our reproductive science. This is one of my favorite all-time papers from Emily Martin about the egg and the sperm and the way science has constructed a stereotypical romance around chromosomes and what we think of and talk about as sex chromosomes, talking about strength, talking about just about anything, to really think carefully about how are these kinds of stereotypes and biases maybe finding their way into our interpretation. So I just want to finally offer a little bit about Sarah Richardson's recent work on sex contextualism as a way of moving past the male-female binary in our design entirely. And she argues that if we conceptualize sex simply as maleness and femaleness, then we will operationalize sex as comparing males and females. And Sarah is attentive to the fact that, for biomedical researchers, for health researchers, there are material realities that we need to deal with about sexual reproduction, tissues, cell types, so on. And we need to attend to that variation related to those functions. But at the same time, we really do need to do a better job of not overemphasizing sex differences, because that kind of stereotyping can risk improper treatment of patients who don't conform to a group mean, as we've seen in the examples I presented above. So for sex contextualism, she said the alternative she offers is, instead, if we conceptualize sex as variation produced by sex-related variables, then we'll operationalize sex as material factors that vary among and within sex subclasses. And in that way, she says, her idea of sex contextualism doesn't start with the assumption that the basis for our design should be male and female. It doesn't necessarily assume that we should be treating sex category as a variable in itself. But it also means that there's no single recipe. There's no single way that sex and sex-related variables ought to be operationalized. It's always going to depend on the research context. What is the question? What are the sex-related factors that are plausibly associated with the phenomenon of interest? And how can we design the experiment in ways to manipulate measure, modify, and examine those pathways that is appropriate to the experimental setting. So it puts the onus back on the researcher to think about what is relevant here. What is the most important thing in the work that I am doing, as opposed to going to some umbrella effort to cover all possibilities. And for gender contextualism, Sarah doesn't talk about this in the paper itself, but I think it's very similar. If we remember that gender is more than just gender identity, and if we think about the ways that gender can be translated into the biology of the body, we can ask ourselves as researchers across the whole spectrum, from cellular work all the way through clinical work, what components of gender, what factors associated with gender might have an impact on the pathways that are relevant for my work, and can we model those in the work? Can we measure those? Can we incorporate that in the design of the study? And I think it's also important at the same time to think about the kinds of gender associated confounders that might exist. So for example, when people are doing work on PTSD in military contexts, if they find that PTSD is more common among men with a history of military service. Is that because because of maleness? Probably not. It may well have to do more with the fact that the occupational role played by men, they're more likely to be placed in direct combat roles and have more exposure to the kinds of things that would cause PTSD. So thinking about what are the mechanisms? How might gender get into the body, and how might gender associated factors moderate our exposure to the relevant triggers? So this is just a summary of the last few slides. I think if we want to transcend the male-female binary, we can recognize and embrace the complexity of sex and gender. We can think about changing our data visualization practices, paying attention to the statistical tests that we apply, and acknowledging that sex and gender category is different from other kinds of variables. We should be conscientious and critical about our rhetorical practices, and we should start to shift our thinking about factors rather than categories. And so I just want to acknowledge a few people that I've worked with over the last number of years who've had a profound impact on my thinking and my work in this area. And thank you for your attention, and invite your questions.
Well, thank you, Dr. Ritz, for that wonderful talk and just giving us an overview of the complexity of integrating sex and gender considerations in our work. And, I see that one of the last questions was how to incorporate this into the research. Can you provide some examples?
Yeah, I mean, I'm going to borrow some of the ones that Sarah has talked about in that sex contextualism paper. One of the ones that she talked about that I think is especially illustrative is one where there had been some findings about estradiol and wound healing, and that there was some evidence to suggest that estradiol was conducive to wound healing. And some of the earlier studies that had done that had been, male-female comparisons, and, they found blah, 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 blah. Her example of some of the follow-ups that were done about that were the ways in which people began to acknowledge, okay, estradiol is the mechanism here. So how do we design our study to focus on that mechanistically, rather than just doing a male-female comparison again? So, and there have been a number of studies in that area, and the way that they've approached it has differed depending on the context. So some of the mouse studies, for example, we're looking at doing oophorectomy and gonadectomy to remove gonads in animals at various points in time relative to the wound model, and examine the impact of estrogen and estradiol specifically, or applying estradiol to the wounds directly, or to administering it to the animals to achieve a certain concentration. In some of the studies that we're looking at, human studies, they were grouping individuals based on their measured estradiol levels, not based on their sex category, but on their actual measured estradiol levels, and seeing relationships there. So once they identified that estradiol was the pertinent mechanism, they were able to shift the experimental design to focus on the mechanism rather than the category. She offers a number of other examples in that paper, so I highly recommend that for some good reading.
Thank you. And I want to thank you also for providing the references, to important references, because I know that's often a comment.
Yeah.
Talks, how do we do this? It's
Yeah.
hard. And as somebody, this other comment, that sometimes the challenges is what I always get is sample size. How, I have to have a certain sample size to integrate all these intersections, all these considerations. So
absolutely. I mean, as a basic researcher for myself, coming from from laboratory studies, like when you start getting a study of 30, 40 mice, depending on what you're trying to, the samples you need to acquire and the processing you need to do, you can't, it becomes unwieldy and undoable when you exceed a certain size. So that's why I think, the focusing on mechanism per se can actually enhance our ability to discern these things more carefully, can make it more feasible to do it with small sample sizes and with manageable, feasible experiments. Because if we are saying, if we recognize that there's a lot of overlap in the distributions between male and female animals, for example, in order to probe that experimentally, you need a much larger sample size to do it if you're just doing this very crude, simplistic male-female comparison. Whereas if you understand more carefully what the mechanism is and design the experiment to the mechanism, you can probably achieve similar insight or better insight into the phenomenon with a reasonable sample size.
Mm-hmm. Well, I'm not seeing any more questions in the chat. I wondered what you thought about the Sager guidelines.
Yeah, so the Sager guidelines, there's some of those, so there's policy issues that have been driving a lot of thinking. And for those who don't know, it's a group of journal editors that had come up with guidelines for reporting about sex and gender in publications and manuscripts. And I mean, I think in general that they're good. In general, they certainly push us in the right direction. I do think they tend to be, again, they're very oriented around thinking of sex and gender category and these binaries. And I think, but I do think they have, a reasonable amount of scope in there for recognizing different forms of research where this might, where some adaptability or some different approaches might be appropriate. So for example, when we're talking about cell culture research, the Sager guidelines don't say you must include male and female cells. They talk about, make sure you identify the origin of those cells and if it's known, sometimes it's not even known, or to justify the use of a particular cell line. One of the early triggers for me investing in this work was that I was at giving a talk about this question about cell lines and transformed cell lines and how you can't just get male HeLa cells. It doesn't exist, but that doesn't mean that HeLa cells aren't a reasonable and useful experimental tool. And one of the attendees, who is a feminist scholar of science, but who doesn't come from a scientific background, had said, well, if you're doing, at the time I was working in A549 cells, which are lung cancer derived cells from a male donor back in the 60s or whatever. And she said, well, if you're doing all your work in these male cells, you're not representing women at all. What are the implications of that? And I don't remember, I stumbled through an answer at the time, but after the fact, I thought, well, we're not representing men either. These cell lines are not representing an entire group of people. A549 cell lines came from a man, yes, but he had cancer. These are cancer cells that have been living in a dish for 40 years. These do not represent men. So I think we need to, I think it's more of an additional argument for the ways that restricting ourselves to male versus female, men versus women, limits our thinking about how we can, about what we're doing, limits our understanding about what we're actually doing and how we might do it differently.
Well, thank you very much. I know we've gone past our time, just shows the interest in this talk, and want to thank you for coming, and also thank our IDEA team, Margaret Kenyaju, Catherine Karakasis, and Azadeh Yadoli for also supporting this talk and having it as and thinking of you in terms of presenting today.
Well, thanks very much for having me. I really appreciate it.
Okay, thanks everyone.